What do cystic fibrosis, a form of dementia similar to Alzheimer's disease, and sickle cell anemia have in common? They are all the result of a point mutation, which is when bases are changed in a person's DNA. But in order to understand that definition, we'll need to talk a little more about genes, bases, and mutations. Let's get started. Your DNA, or deoxyribonucleic acid, is, to put it simply, the blueprint for how to make you. It gets nicely packaged into chromosomes, which can be found in your cells. There are sections of your chromosomes called genes, which code for specific amino acids. When you get a bunch of amino acids together, you have a protein, and proteins serve many, many functions in your body, from making muscle to producing insulin. If you were to zoom in on your DNA, you'd see the following bases, or parts of the nucleotides, that make up your DNA. Adenine, A, thymine, T, guanine, G, and cytosine, C. These bases pair up. Adenine and thymine pair together, and guanine and cytosine pair together. Often, you'll see the bases simply written as letters. As you can probably guess, adenine is A, thymine is T, guanine is G, and cytosine is C. Okay, now you're ready to look at the definition of a point mutation again. When one of the bases gets changed, you have a point mutation. A point mutation can result in the wrong amino acid getting produced, which alters the protein. Remember, a protein is made when a bunch of amino acids are linked together. And if the protein gets altered, you can have all sorts of problems. Let's take a look at the different types of point mutations so you can get a better idea of what can happen when your DNA gets mutated. We are going to focus on the following point mutations: frameshift, silent, nonsense, and missense. Let's start with a frameshift mutation, where there's an insertion or deletion of one or more bases. In other words, bases get added or removed. Why would that matter, you ask? Well, it takes 3 bases to code for an amino acid, and this is called a codon. For example, if you have cytosine followed by 2 adenines and you transcribe it into RNA, you get GUU, which codes for the amino acid valine, abbreviated VAL. Remember, you have to transcribe the DNA to RNA first. Also, thymine is replaced by uracil in RNA. You can use a codon chart to figure out the amino acids. When one base is added or deleted due to a mutation, it shifts the bases and causes a frameshift mutation. Let's take a look at a section of DNA to illustrate this. Say you have the following DNA: DNA GATCTC AAA, which transcribes to mRNA CUAGAGUUU. Before the mutation, the DNA transcribed to CUA coding for the amino acid leucine, GAG coding for glutamic acid, and UUU coding for phenylalanine. So pretend you have a deletion of the adenine, A, out of GAT. Everything is going to shift. Now you have DNA GTCTCAAA, mRNA CAGAGUUU. Instead of leucine, you will get glutamine. Instead of glutamic acid, you get serine. And instead of phenylalanine, you may not get any amino acid, since there isn't a complete codon. You can see how a frameshift mutation can change all of the amino acids in the protein, thus changing the protein itself. Cystic fibrosis is an example of a frameshift mutation. It's a genetic disease that causes the production of excessive mucus that damages the lungs and digestive system. It is caused by a deletion that results in an amino acid not getting produced. Next, let's check out a silent mutation. This is when a point mutation occurs, but it doesn't change the organism. It may be a mutation that doesn't occur on a gene, so the codons aren't affected. Or it may be code for a similar amino acid that still functions correctly. In a nonsense mutation, a codon is altered in such a way that it leads to a premature stop before the protein is done. There are a few codons that indicate the amino acids can stop being added and the protein is done. Let's say you had the codon ATG, which transcribes to the codon UAC for the amino acid tyrosine. In a nonsense mutation, the guanine, G, might get replaced by thymine, T, and give you ATT, which transcribes to UAA, which means stop. The result is an incomplete protein, which may be totally ineffective. There is a rare disease called familial British dementia, where the protein that should be produced is stopped short due to a nonsense mutation. The disease usually appears when the person is around 60 and results in dementia similar to Alzheimer's disease. Finally, a missense mutation occurs when a base is changed and replaced with another. This may result in a different amino acid being produced. For example, sickle cell anemia is a disease where the red blood cells change into a sickle shape, which results in pain, strokes, and infections. In most cases, sickle cell anemia is caused by a missense mutation, where an adenine is replaced by a thymine, and instead of the amino acid glutamic acid, you get the amino acid valine. The change in amino acids results in the sickling of the red blood cells and all of the associated problems that go with this change. All right, let's take a moment to review what we've learned. Who knew small changes in your DNA could cause so many problems? But as we've seen, point mutations, or changes in the bases of your DNA, often do cause problems. Remember, your DNA is made up of 4 bases: adenine, thymine, guanine, and cytosine. Changes in the order and number of these bases can result in different point mutations, including frameshift, silent, nonsense, and missense. In a frameshift mutation, one or more bases gets deleted or inserted. This alters the protein that's produced. For example, some forms of cystic fibrosis are the result of a deletion of a codon, which involves 3 bases to code for an amino acid, resulting in a missing amino acid. A silent mutation is when something gets changed on the DNA, but it doesn't impact the organism. Another point mutation, a nonsense mutation, occurs when the DNA codes for a stop instead of another amino acid. The result is an incomplete protein. An example here is a form of dementia known as familial British dementia. Finally, a missense mutation occurs when one of the codons gets replaced by another, resulting in a different amino acid. An example here is sickle cell anemia, when a different amino acid is produced, resulting in defective red blood cells.